Hello, my name is Brigid Driscoll, and I'm a senior at UMass Amherst studying mechanical engineering. My thesis is on the dangers of CRISPR gene editing, and specifically an analysis of the dual-use concerns regarding gene editing, the potential for it to become a bioweapon threat, and current protocols surrounding DNA accessibility. The field of synthetic biology has grown significantly with technological advances such as gene editing. This field has seen remarkable advancements in the past decade, with technologies such as CRISPR developing at a rapid rate. CRISPR technology allows scientists to easily alter DNA sequences, promising advances in curing specific diseases such as HIV and AIDS. CRISPR technology is revolutionary because it's easier to use and much more simple than other gene editing technologies. There are also many future advancements that scientists are always progressing with the help of CRISPR, such as editing certain species' DNA to prevent extinction, making genetically modified foods healthier, and eradicating deadly diseases in animals, such as malaria and mosquitoes. However, there are also many risks associated with using CRISPR technology. When scientists edit somatic cells, the edits only apply to the individual. However, editing germline cells affect all cells in the body, including reproductive ones. This means that the changes to the individual's DNA will be passed down through generations to their children as well. This brings up many concerns, such as the morality of changing an individual's DNA before they are born to consent. It also raises the concern for unknown effects that may not show up for years to come. So there's many arguments against editing the DNA of human embryos before they're born. The main concern is where to draw the line between disease treatment with diseases that are already existing or may exist in the baby versus enhancing their physical traits, such as appearance or intelligence. This brings into question, could gene editing create what some may call designer babies, or babies that are biologically superior due to modifications before they're born? This process also could create unintended consequences that may not present themselves for years to come. Because of all these effects, the National Institute of Health and the FDA have banned funding for germline editing research. The first successful birth of a modified human embryo was in China in 2018 when a scientist birthed two twin girls who were HIV resistant. Although this was a huge breakthrough, there was universal backlash to the experiment. It was deeply flawed, both in testing and in the scientific report, and scientists worldwide agreed that the experiment was very premature as well as unethical. The research that the scientists conducted was censored in China across all social media platforms, and the scientist was arrested. This case study reveals that gene editing research is likely being conducted behind the scenes in many countries, despite the rules that are set in place currently about biosafety. There's also a growing market for do-it-yourself gene editing kits that individuals are able to purchase online and edit DNA at home. Although this market is very innovative and inspiring for future researchers in the biotechnology field, the ease of CRISPR technology and accessibility of DNA creates concern for biosafety, as people can easily purchase pathogens online and manipulate them as they wish into a bioweapon. I'm currently surveying public opinion about gene editing and CRISPR technologies. My questions include the individual's familiarity with CRISPR, their opinion about the morality of editing human embryo DNA, if CRISPR should be classified as a weapon of mass destruction, and their opinion about if current laws surrounding gene editing research should be more strict. My main angle when analyzing the data collected has been to study the effect of age on public opinion. Since CRISPR is such a new technology, it's important to analyze each age group's familiarity with the technology as well as their opinion about its potential threat. So the graph on the left is from 2018, and it highlights the public support of gene editing when it comes to treating or preventing a specific disease in a baby. With the constant advancements of CRISPR, public opinion is constantly changing regarding the morality of gene editing, and public opinion has changed significantly since 2018. My research is still ongoing, but my findings so far have supported the fact that the younger the individual, the more likely they are to support gene editing in human embryos. In conclusion, public opinion has a very large impact on the laws and the protocols implemented in a country. If the public doesn't support a stance, the government will most likely follow this direction in order to appease the general mass. My research so far has supported the hypothesis that laws regarding gene editing research, as well as DNA accessibility, must be more strict in order to prevent technologies such as CRISPR from becoming a bioweapon threat. Thank you for watching and have a great day.